Good morning, everyone. Today's experiment is based on bright field and polarized light microscopic techniques. What is a microscope? It is a laboratory instrument used to examine those objects that are not usually visible through our naked eye. If we break this term, then it then here micro means small and the scope means to view. So microscopy means to view small objects. The most common microscope is the optical microscope, which uses lenses to refract visible light that passes through a thinly sectioned sample to produce an image. This is the basic classification of the microscopes. Microscopes can be of two types, broadly classified into two types: simple microscope and compound microscope. In simple microscope, there is only one lens, and in compound microscope, more than one lenses are used. Compound microscopes can further be divided into light microscopes and electron microscopes. In light microscopes, light is used for illuminating and visualizing the specimen, and in electron microscopes, electron beam is used for visualizing the sample. Light microscope can further be divided into bright field microscopes, dark field, phase contrast microscopes, and fluorescence microscopy. Similarly, electron microscopes are of two types, that is scanning electron microscope and transmission electron microscope. This is the basic diagram of a optical microscope. These are few parts and their functions. First of all, there is a eyepiece. It focuses the image from the objective lens. This is where image is formed. Then there is objective lens, which makes a clear image from the specimen. And there are two focusing knobs, which is used for decreasing or increasing the focus according to our requirement. Then there is stage on which the specimen is placed. Then there is condenser lens. The function of condenser lens is that it converges the light onto the specimen. Then there is arm. It has a light illuminator, which is the light source, and then an aperture diaphragm, which controls the diameter of the beam of light that passes through the condenser. There are three main optical pieces in compound light microscope. First is condenser lens, then there is objective lens, and then ocular lens. The main function of the condenser is that it converges a beam of light onto the object or the specimen that we are visualizing. Then there is objective lens. It forms a magnified and inverted image of the object. And then ocular lens further magnifies the image that is formed by the objective lens. So the total magnification power of a compound microscope is the product of the magnification of the objective lens and the magnification of the eyepiece. The bright field microscopy is the simplest type of optical microscope. Uh, the name of then the term bright here in bright field microscopy is given so that because the object is dark in this kind of microscopy and the background is bright. Here the light is transmitted through the sample and the contrast is generated by the absorption of light, because of which the specimen is visualized. Here, the specimen can seen directly by this microscopy should possess color or some properties, because this specimen only this microscopy only visualizes the objects which are colored or stained. So this is one of the limitation of the bright field microscopy, that if we need to visualize any specimen, then we must stain it before, or it should contain natural color, for example chlorophyll cells. Another limitation of bright field microscopy is that it is a it has low contrast for weakly absorbing samples and low resolution. Polarized light microscopy is another kind of light microscopy. Uh, what is polarized light? Polarized light means the light which have the components wave components which are traveling in the same direction. Any light that vibrates in more than one direction is called unpolarized light, whereas a light wave that vibrates in single direction is called polarized light. Polarized light microscopes work by converting unpolarized light to polarized light. So for this purpose, a specific kind of polarizer is used in this kind of microscopes. Unpolarized source is converted into polarized light before it hits the sample or the specimen that we are visualizing. This polarized light then falls on the specimen, which generates two wave components. And these wave components are at right angles to each other, and these are called ordinary and extraordinary light rays. These light waves then go through the specimen and then combine through the destructive and constructive mechanisms of interference. And as a result of which, a high contrast image is generated. This is the diagram of the polarized light in microscope. The only difference between a bright field microscope and the polarized light microscope is that here our polarizer is introduced for converting unpolarized light into polarized light. This is the difference between the images captured by bright field and polarized microscope. And in a polarized microscope, images is much more clear, and it can be clearly seen that the resolution is higher as comparison to bright field microscope. These are the samples that we'll be visualizing today. First one is the chocolate syrup, and then sugar particles dissolved in water, and then oleogel.
There are four main parts in a microscope: one illumination unit, then the condenser, this one, then you can go for this eyepiece, sorry, objective lens, objective lens, then your eyepiece. Okay, so to move this sample, we have this sample stage. Okay, we can move this sample stage in three coordinates: x, okay, then y, then for the z movement here we have this knob, this bigger knob to move the sample, but not vertically, for the coarse adjustment, and this small knob is for the fine adjustment. वो vertically ही movement होगा, but for the coarse and the fine adjustment ये आपको अगर ज़्यादा movement करना है, ज़्यादा एक रोटेशन में अगर आपको ज़्यादा ऑफ या डाउन में जाना है, तो यू कैन यूज दिस वन. एंड फॉर द पर्पस ऑफ व्हेन जब आप फोकस लाना चाहते हैं पर्टिकुलरली फील्ड ऑफ व्यू में, तो आपको ये जो स्मॉल नॉब है इसको एडजस्ट करके आपको वो फोकस में लाना होता है. ओके, सो these are the few basics that have been we also covered in the PPT, right? Now coming to the bright field and your polarized light microscopy. In the polarized light microscopy, we use filters, polarizer filters, polarizer, right? So if you recollect it, in the presentation we have shown you two polarizing filter that has been used to make a bright field image into your polarized image, right? So this is the polarizer that has been that is going to be placed near to your illumination unit, and there is another polarizing filter that has been placed in your objective lens. Okay, so this is called analyzer and this is called polarizer. जो हम illumination unit के पास रखेंगे that is called your polarizer, और जो objective lens के पास पर जो है that is called analyzer. Okay, so the purpose of these two is well explained, right? I think you might have got the use of these two seats, why we actually use these two seats. So basically, I'll just again make you understand that this can be adjusted such that you can make the EM wave जो जा रहा है आपका will make the electric field and the magnetic field in a single phase, single plane you can say, right? जो usually हमारा क्या रहता है 90 डिग्री में जो phase difference रहता है we can make it into a single phase, उनका जो phase difference होगा that is be zero, right? ये for that reason we are using this, and once that normal electromagnetic light wave got converted into polarized light wave, it will got interacted with your sample over here, and it will enter this objective lens, and after that this analyzer will come into picture. Okay, अगर कोई भी सैंपल के अंदर आपका कुछ भी पार्टिकल या कंपाउंड या मोलिक्यूल जो भी है बोल सकते हैं, वो अगर इंटरेक्ट कर रहा है पोलराइज्ड लाइट के साथ, मींस व्हाट, इट विल चेंज इट्स फेज, फेज का जो डिफरेंस होगा दैट विल बी गेटिंग डिटेक्टेड बाय योर एनालाइजर, एंड वो आपका either you can see it through the eyepiece or you can use the sensor or camera over here and you can capture the image or you can see the things, right? Without no, right? So let me turn it on. This is salicylic acid dissolved in propanol, so write it down. पहला आपका जो सैंपल है वो salicylic acid है, so we'll put it over here. Okay, so we'll see. देखो इसको जब भी आपको फाइन एडजस्टमेंट, जब भी आपको फोकस करना होगा, यू हैव टू यूज दिस फाइन एडजस्टमेंट फोकस. ओके, these are the crystals for your substance. बढ़ा दे. Is it okay? No, I captured it. तो ये फोटो आपको मिल जाएगा वो रेकॉर्ड बुक के लिए, ये बस इंस्ट्रूमेंट का फोटो लेके चले जाना, ठीक है? तो now I will see the polarized one. Can you see the difference? The different part of the crystals is glowing in a different color also, right? This is due to the scattering as well as no diffraction होगा. मतलब diffraction होगा. लाइट आ रहा है आपका नीचे से, आपका ऊपर सैंपल है जो डिफ्रैक्ट कर रहे हैं, उसी के वजह से वो ग्लो कर रहा है, राइट? तो अभी क्या हुआ in this micrograph, the background is totally black and the crystals that are there in the sample is only blue, right? तो ये फिर डार्क फील्ड हो गया ना? डार्क फील्ड में अलग आएगा, डार्क फील्ड देखोगे नेक्स्ट, ठीक है? ये लिखो, ये आपका कहाँ से है, 13 से शुरू है, 13 और 14 आपका जो सैंपल है वो लिखो, आपको लिखना पड़ेगा क्योंकि हम ये वाला इमेज ही शेयर करेंगे आप लोगों के साथ, आपको उससे सिलेक्ट करना है आपका जो इमेज है, ठीक है? दिस इज ओलियोजेल. ओके, ओलियोजेल इट्स ए मिक्सचर ऑफ ऑयल एंड सम वैक्स क्रिस्टल है, एक्चुअली वो भूल जा रहा हूँ मैं. ओके, सो इन दिस ओलियोजेल वी हैव wheat germ oil and cocoa butter. Okay, so we'll see. I'll tell you the difference. अगर आप यहीं पे देखेंगे, देखिए दो दो मेक इट लिटिल बिट, यहीं पे दो फेज आपको दिख रहा है. देयर आर सम needle needle kind of structures and there is a big background में एक है जो कि एक एक पूरा यूनिफॉर्म ये दिख रहा है, राइट. सो दिस क्रिस्टल्स, दिस जो निडल शेप का जो है, दिस इज योर कोकोआ बटर का जो उसमें जो कंपोनेंट्स है उसका है, एंड द बैकग्राउंड वन इज योर ऑयल फिल्स. ओके, सो उधर जाना थोड़ा, वो जहाँ पे बैठता हूँ ना उधर चार्जर रखा हुआ, थोड़ा लेके आ जाओ. Okay, so we'll capture this once again. We'll see कि कैसा आएगा अगर हम उसको अगर पोलराइज्ड में दिखेंगे तो. दिख रहे हैं द फेज जो बैकग्राउंड में था दैट इज टोटली गॉन. अभी जो क्रिस्टल्स है दैट आर वही बस ग्लो हो रहा है. ठीक है, हो गया. ये लिख लो आपका जो इमेज है, ये इसके लिए मेल्टेड चॉकलेट. चेक इट. ठीक है, चॉकलेट में तो बहुत सारे चीजें रहती हैं, जैसे कि कोकोआ पाउडर भी रहता है. ओके, लाइक वाटर कंटेंट कुछ रहता होगा, देन शुगर, देयर आर मेनी थिंग्स दैट आर देयर. अभी हम दिखेंगे कि ये इसका करस्पॉन्ड, ये उसका हो गया ब्राइट फील्ड. इफ यू गो फॉर इट्स, अभी देखो यहीं पे कुछ क्रिस्टल क्या है, यू विल नॉट एबल टू डिफरेंशिएट. ओके, व्हेन यू पुट दिस, गो फॉर दिस polarized one, you can see the difference over here. दिख रहे हो ये वाला पार्ट्स जो है ज्यादा ग्लो नहीं कर रहा है ऑल्दो, बट इट्स सेपरेट हो रहा है, glowing little bit, right? These are the sugar crystals that have been there in here. Okay, दिख नहीं रहा है बट दूसरा जगह पे ले लेते हैं. ये वाला ले सकते हैं. देखो यहीं पे दिख रहा है है ना. उसका कॉरेस्पॉन्डिंग ये ले लो. ये अच्छा लगेगा इसमें. रुको मत लिखो, उसको दूसरा लेते हैं. ये दे देना.
सर ये नया स्लाइड
ठीक बना दिए ना. डोंट थिंक सो. ठीक बना है।
ठीक है सर।
उसमें आ रहा है। नहीं ये ठीक है, नहीं आएगा इसमें। इसमें ये देते हैं ना, क्या है, नहीं तो उसको ही दे देना जो पहला वाला आया था, ये दोनों को दे देना। ये दिस नहीं, ये तो अलग जो पोजीशन में मैं ले लिया था ना, नहीं यही है, यही है क्या? हाँ यही है ना, तो इसको दे देना, ठीक है 1780, ठीक है। ओके, एनी डाउट?
ये क्या है?
ये वो मल्टीप्लेट रीडर का डिश वॉशर, मल्टीप्लेट डिश वॉशर, मल्टीप्लेट वॉशर ऐसा कुछ है। ओके, नेक्स्ट ग्रुप को बोल दो जय जय। अपने आप ये